And here we go. We're going to look in this video at protection for the brain. First, the cartoon. CAT scan. Okay. Let's look at how the brain is protected. I think we can all agree the brain needs to be protected pretty vigorously given its major role in all of our functioning as well as protecting the delicate neural tissue. So let's look at some of the ways the brain is protected. One, of course, is rather self-obvious. The cranium, the bones of the skull, are going to provide major protection for the brain. But then we're going to look at some other mechanisms that are somewhat unique to the brain. The reason I qualified that as somewhat unique is we're going to see our first ones are actually the meninges, and we should be familiar with meninges. We talked about the dura mater, the arachnoid mater, and the pia mater with the spinal cord. Good news, they're all going to be the same meninges in the same order with just one slight variation in the brain for the cranial meninges. So let's start with the dura mater. The dura mater actually has two distinct layers, and this is the difference in the meninges in the spinal cord in the brain. In the brain, the cranial meninges, we're going to have a periosteal layer and a meningeal layer. So it's separated into two separate tissue layers. They're both part of the dura mater. It's just they have a division here we don't see in the spinal cord. The periosteal layer is going to be directly underneath the floor of the skull. So it's directly connected. Periosteal refers to position next to bone. So we're going to find the periosteal layer directly underneath the cranium. And then attached to that in lots of places, we're going to find the meningeal layer. There's going to be a few spots, though, where we find that they separate. Again, here is the periosteal layer, here's the meningeal layer, but notice what they do, they separate here, and we end up here with this space. This is going to be what's called a ductal venous sinus. This happens to be the superior sagittal one, but this sinus here is going to be a cavity or a space. In this case, this is where venous blood from the brain is going to move into this space. And eventually, this blood is going to go into your internal jugular veins, eventually go to your heart, and be sent around the body. So most of the time the layers are going to be stuck together, but here they're going to separate and create this space. The other significance of the meningeal layer is if you kind of follow along here, notice this layer separates and actually goes down into the very superior part, or the most superficial part of the brain, and actually forms this called falx. This happens to be the falx cerebri. There's also a falx cerebelli, but these falx are little false partitions or little walls that are going to come into the very exterior part of the brain and hold it into position. Why does the brain need to be held in position? The brain is basically floating inside your cranium, and we don't want it to be sloshing around too much. So the falx, the meningeal part of the dura mater, actually hold the brain somewhat in position so it doesn't slosh around too much. Okay, so that's the dura mater. There's going to be a subdural space, and it's going to be a serous space, which means there's going to be a slight greasing fluid there. The next layer, the next meninge, is the arachnoid, and we've seen the arachnoid before. It's going to be a thin, whispery layer that it's a little hard to see on this diagram, but it's going to be right there, I believe. It's going to serve as a somewhat of a chemical protection. Then below that, we're going to have the subarachnoid space, and that's what looks like all this stuff in here. It looks like little hatchings. That subarachnoid space is going to, of course, also be where we're going to have CSF, cerebral spinal fluid, as we had in the spinal cord. So the cerebral spinal fluid, and we'll get to that more in a moment, is going to be flowing into the subarachnoid space. Also notice we have blood vessels, and as you can imagine, they're not well protected here because they're in this wispy space with some fluid in it. And if these are ruptured in the form of trauma, you can have a subarachnoid hemorrhage, and that can be very serious very fast. So we'll talk about that more when we talk about our traumatic brain injuries. The final layer, of course, is the pia mater. It's going to be a very thin layer. It will have some blood vessels associated with it, and it's going to be directly on the brain itself. Here's just a slight purple line that you see here. So these represent some major protective layers. All of these layers are going to be contiguous with, or they're going to join up with, the spinal cord dura, spinal cord meninges rather. The periosteal layer isn't. That's only going to be associated with the skull. But the dura mater, the arachnoid mater, and the pia mater are going to eventually form and connect with the spinal cord meninges as well. So let's talk more about the fluid, the cerebral spinal fluid, that we've already addressed a little bit in class and a little bit in the previous slide. We have the cerebral spinal fluid, as you recall, formed by some of our neuroglial cells. These are ependymal cells that are going to be found in these spaces. These are the ventricles. Let's come back for a second. The ependymal cells are going to be found in, let me get a typing here, in the choroid, plexus. This is basically just where you have a bunch of ependymal cells together. They form this region, and this is where you're going to produce cerebral spinal fluid. Recall that essentially it's a filtered version of your blood. So in the ventricles, in these fluid-filled spaces in the brain, you're going to have choroid plexuses. You're going to have ependymal cells in there, and they're going to produce a cerebral spinal fluid. The ventricles, you have 4. The first 2 are called the lateral ventricles, and you see them here. When you're looking at a brain in lab, for example, you may see like an open space or a gap. That was where the ventricles were. Okay, so the first 2 are just called lateral ventricles. The third ventricle, we said in class, is associated with the thalamus, and then it drains into a small space. You can see it better over here, the fourth ventricle. And from there, cerebral spinal fluid can go a couple directions. It can go then down the central canal, which is through the spinal cord, or some of the fluid, cerebral spinal fluid, is going to flow around the brain through the subarachnoid space, and eventually, and we should probably go back and do that in a second, we'll see where it goes in just a moment, but the key idea here is having these fluid-filled spaces in the brain provides some protection, provides some buoyancy, and allows the brain to have some degree of stabilization inside the cranium. Let me go back for just a second to, not doing that, to where does the cerebral spinal fluid go? Actually, I think you see it on the next slide, so let's go ahead. Okay, so this shows you, let me erase, this shows you the flow of cerebral spinal fluid, and you can't see it well. I had a cool diagram, it's not working, but the fluid is going to flow from the lateral ventricles around the third ventricle through the fourth ventricle, and then again some of the fluid is going to flow through the central canal of the spinal cord, and some of it flows around the brain. And what you don't see here, so I do need to go back, is where does that fluid go? Some of that fluid, and this again, this is subarachnoid space, it's going to flow through these arachnoid villus, and cerebral spinal fluid, extra fluid, will be dumped into the bloodstream. Where is this blood going again? The internal jugular veins, and eventually back into the bloodstream. And you're thinking, wait, we can't do that because we can only have blood in the bloodstream. Remember, the cerebral spinal fluid is basically a blood derivative, so there really is no issue with putting it back into the bloodstream. Oh, there it is. It's kind of working now. You can kind of see it. So this is showing you the flow of cerebral spinal fluid. If you look very carefully at this MRI, you can see a little bit of flow here, and you can see some flow down the spinal cord. And again, what is the CSF doing? It's protecting, it's nourishing. It actually provides some chemical protection for the brain as well. You need to have a very stable chemical environment around the brain, and the CSF helps do that. What if those delicate tubes, those delicate connections between the ventricles clog up? You can see that in this. This is actually a case of hydrocephalus. Here you see a normal brain, and in this section, looking at transverse section, of course, we're seeing the black spaces represent the ventricles. That's how big they should normally be. This person over here has some issues. They have these spaces that are much larger than it should be. We have fluid in some ventricular spaces that's not flowing out. What's the problem? The fluid is being produced, it's being generated and filling up these spaces and not flowing. This thing on the outside, this is your cranium, it has no give. It is not going to budge. So as the fluid fills up, it begins to push on the brain tissue and can cause some serious and permanent damage. So for hydrocephalus, we're going to have to drain that cerebrospinal fluid out before it creates too much pressure. Interestingly, with kids, we can kind of get away with it, so to speak. What's different about a child below the age of 1 and an adult? A child is going to have unfused skull bones. So if a child has hydrocephalus and there's extra fluid or a blocked shunt somewhere in their network of cerebrospinal fluid flow, then they can accumulate some fluid in their brain. Their skulls can expand because they're not welded together yet by it, not infused by sutures. And so you can see in this, there may be no long-term consequences. They will just put a shunt behind the ear, drain out some of this fluid, and try to treat the cause of that blockage. And if so, then the child will be fine. For an adult, hydrocephalus is going to be far more dangerous because a lot of cerebrospinal fluid will actually eventually push the brain against the skull and cause serious and permanent damage. We'll leave off, let me get out of the way here, we'll leave off with one other form of protection for the brain, and that is the blood-brain barrier. We've actually already talked about this because another neuroglial cell, the astrocytes, are involved here. But let's just start with this. Why do we care? We have to maintain a really normal, stable chemical environment around the brain. Otherwise, those neurons that are highly sensitive to ion changes, too much potassium, too much sodium, will cause them to fire. That may interrupt the flow of neurotransmitters. So we have to be very careful that we don't actually put anything into the brain that might chemically alter it. So astrocytes and endothelial cells of the brain, basically cells that line the capillaries, are going to form this blood-brain barrier. It's not a barrier as in a wall. It's simply where, and it's a little hard to see here. Let me get out of the way again. Let me disappear. You see that the astrocytes here are going to wrap around and protect the, it's a little hard to see on here, the astrocytes here are trying to actually protect around these blood vessels, and they prevent things from simply diffusing out. And since they don't diffuse freely out, things that, let's say, pathogens, viruses, some chemicals that would normally just come out of the blood because they have a chance to, aren't going to come out in the brain. So the astrocytes are helping to form this barrier. And you see here a little policeman. You're too highly charged, you're too large, and you're not lipid soluble. Those things are not going to be allowed to enter the brain. There are some spots where you do not have these astrocyte protections, and this might be where some pathogens actually do sneak out of the capillaries and sneak into the brain and cause diseases and cause problems. But let's go back and review for 1 second. So our major protections here: cranium, the meninges, the dura mater, arachnoid mater, and pia mater. We also looked at cerebral spinal fluid, how it physically protects. It gives you a buffer, gives you a buoyancy, gives you some bounce, if you will, to protect the brain, and also chemically protects the brain by maintaining a fluid, a solution around the brain that's chemically stable. And then we also considered the blood-brain barrier. In our next video, we're going to look at traumatic issues with the brain. What happens to the brain when someone has a concussion or when someone has, for example, a hematoma. All right, that's it for this one. Thank you.